Good morning everyone. Today we are going to discuss chapter 2 of class 12, that is sexual reproduction in flowering plants. And we know that all flowering plants show sexual reproduction. In this chapter, let us understand the morphology, structure and processes of sexual reproduction in flowering plants, that is in angiosperms. And as we are talking about flowering plants, let's have a look at the structure of the flower first. These are petals and sepals which are called non-essential parts of the flower. It does not mean that they are not essential, but they are not directly involved in reproduction. And the essential parts are pistil and stamen. Pistil consists of stigma, style and ovary, and stamen consists of anther and filament. They are called essential parts because they are directly involved in reproduction. Now we are coming to the next topic, pre-fertilization, structure and events. Before fertilization, a series of events occur in flower. The story begins with several hormonal and structural changes which leads to differentiation and further development of floral primordium. What is floral primordium? It is a group of cell or tissue which give rise to flower. And then inflorescence are formed. Inflorescence means modified part of the shoot where flowers are formed. It will bear floral buds and then flowers. In the flower, the male and female reproductive structure, that is androecium and gynoecium, differentiate and develop. And you know that androecium consists of group of stamen that represent male reproductive organ and gynoecium represent female reproductive organ. Androecium is the collective term used for all the stamen present in the flower, and gynoecium is the collective term for carpels of a flower. As we are talking about androecium, let's have a look at the structure of the stamen first. There is a long slender stalk called the filament and the bilobed structure called the anther. और ये जो एंड है जिसे हम बुलाते हैं proximal end, ये जुड़ा होगा थैलेमस के साथ और we can say the basal part of the flower उसके साथ. If we talk about the anther, then it is bilobed structure. आपको ये दो lobes भी दिखाई दे रहे हैं. सो एंथर इज अ बाइलोब्ड स्ट्रक्चर और हर लोब में आपको दो थीका दिखाई देंगे. इट मीन्स इट इज डायथीकस. ईच लोब हैव टू थीका. इट मीन्स टू कंपार्टमेंट्स इन ईच लोब. और एक लॉन्गिट्यूडनल ग्रूव आपको दिखाई दे रही है जो कि पूरे लेंथ वाइज इसको सेपरेट करती है थीका को. अगर हम देखें टीएस ऑफ एंथर को, देन वी कैन से दैट एंथर इज फोर साइडेड और टेट्रागोनल स्ट्रक्चर कंसिस्टिंग ऑफ 4 माइक्रोस्पोरेंजिया located at the corner, 2 in each lobe. इसको अगर हम थोड़ा ध्यान से देखें, एंथर में 2 लोब हैं, हर लोब में 2 थीका हैं. Cells of theca develop to form microsporangium which further develop to form pollen sac और इसी pollen sac में pollen grains produce होते हैं. अगर हम बात करें माइक्रोस्पोरेंजियम के स्ट्रक्चर की, इसका टीएस अगर हम देखें तो आपको दिखाई दे रहा है कि ये सर्कुलर है आउटलाइन में और इसकी जो लेयर्स हैं उसको देखें. It is surrounded by 4 wall layers, epidermis, endothecium, middle layers and tapetum. Outer wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is called tapetum. It nourishes the developing pollen grains and the cells of tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetum cell could become binucleate? अगर हमें मालूम है कि बाई न्यूक्लिएट कंडीशन होनी है, इट मीन्स वहां पर कैरियोकाइनेसिस हुआ होगा लेकिन साइटोकाइनेसिस नहीं हुआ होगा. तो टैपेटम सेल अंडरगोज एंडोमाइटोसिस इन विच न्यूक्लियस डिवाइड्स विद इन द न्यूक्लियर मेम्ब्रेन. मीन्स कैरियोकाइनेसिस हो गया बट साइटोकाइनेसिस नहीं होता और जो कंडीशन है वो मल्टी न्यूक्लिएट हो जाती है. Tapetal cell also have a dense cytoplasm as they are responsible for producing sporopollenin, a protein that forms the pollen wall. So due to high concentration of cell organelles carrying out protein synthesis, the cytoplasm of tapetal cell appears dense. Center में अगर हम देखें तो जो cell हैं, these are called sporogenous tissue and each cell of sporogenous tissue is capable of giving rise to microspore tetrad. Each cell, which means pollen mother cell of sporogenous tissue, is capable of producing new pollen grains. These cells undergo meiotic divisions to form microspore tetrad. As the anther mature and dehydrates, the microspores dissociate from each other and develops into pollen grains. and the process of formation of microspores from pollen mother cell through meiosis is called microsporogenesis. इसका मतलब हमने ये देखा कि ये जो स्पोरोजीनियस टिश्यू का हर सेल है जो कि पोलन मदर सेल की तरह बिहेव करता है, इसमें मियोसिस होता है और इससे एक टेट्राड प्रोड्यूस होता है, मींस 4 सेल, और हर सेल पोलन ग्रेन के फॉर्मेशन की कैपेसिटी रखता है. अगर हम बात करें पोलन मदर सेल तो वो उसकी जो प्लॉइडी है वो डिप्लॉइड होगी और उसमें मियोसिस के बाद जो टेट्राड फॉर्मेशन होगा उसके हर सेल की प्लॉइडी हैप्लॉइड होगी. Inside each microsporangium, several thousands of microspore or pollen grains are formed that are released with the dehiscence of the anther. आप देख रहे हैं यहाँ पे dehiscence हुआ और जो pollen grains हैं वो सारे एयर में आ गए हैं और वो फिर pollination के लिए चले जाएंगे. These pollen grains represent the male gametophyte. So in the next video, we will definitely discuss about pollen grains. Thank you.